Now, in order to do the acid-fast stain, you have to do it after gram staining. The reason behind this is because if the gram staining of our unknown bacteria was gram-positive, the acid-fast stain could be used because it has the ability to show us the differences between the two types of gram-positive bacterial cells. The two types of bacterial cells, gram-positive bacterial cells, include ones that have like a waxy microlytic acids in their cell walls, and gram-positive cells that do not. Before you can start this experiment, you will need to gather some materials. These will include your heat fix slides, which are in this slide box right here. You will need your stain tray. You will need a slide clip. You will also need a carbon fusion stain, which this is going to act as it. In this experiment, carbon fusion is your primary stain. It will stain any bacterial cells that are acid-fast. And then you have your acid alcohol, which will be your decolorizer. You will have methylene blue stain. This is your counter stain. It will stain the non-acid-fast bacterial cells. You will also need a steam bath, which I do not have, but I'm going to find one, something to use for the video. You also need a water bottle for rinsing, bibulous paper, a stain collection vessel, which I do not have, biohazardous waste container, which again I do not have that either. And always make sure that you have your goggles on, a new pair of clean gloves, and your lab coat. And once you have all your materials, you are ready to begin. So to start your experiment, you're going to take your slide out of your slide box, like so. And I'm going to hold on to it with the slide clip so that I can do other things in this experiment. And then you're going to rip out a piece of bibulous paper from your book of bibulous paper, and you're going to put it on top of the smear, like so. And then what you're going to do is you're going to place your heat fix slide with the bibulous paper over the steam bath. Now, this is what I'm going to be using as my steam bath, this thing right here. And I'm going to put it, now if it was a real experiment, you would have a slide rack and all that. So, metaphorically, I will be putting it on the slide rack that's on the steam bath. Okay, I'm going to put my slide clip back down. And once the heat fix slide is on the slide rack over the steam bath, I'm going to apply my first stain, which is carbon fusion. You'd want to saturate the bibulous paper with it. Okay, and then once you've done that, once you've applied the carbon fusion stain, let the heat fix slide sit over the steam bath for about 10 minutes. Okay, so the reason why you need to steam the heat fix slide with the bibulous paper and carbon fusion stain is because the steam from the steam bath will actually drive the stain into the acid-fast cells. After 10 minutes of steaming your heat fix slide, you will remove it using your slide clip, like so, and then you're going to carefully remove the bibulous paper, and since it's now been contaminated, you're going to get rid of it by putting it in the biohazardous waste container. And now we're just going to let the slide cool off. I'm going to put this on the slide rack that is on the stain tray, and then we'll continue on. Now that your slide has cooled off, you're going to rinse it off in a gentle stream of water. But first, you're going to pick it up using your slide clip, tilt it downwards, and then rinse with water. And now don't put your slide down, because what you're going to do now is you're going to apply your acid alcohol. Now remember, this is your decolorizer. You're going to apply about 10 to 15 drops of this to your slide. I'm just going to speed the process up. But once you're done applying the acid alcohol, you're going to rinse your slide down again with water, to get the excess acid alcohol off. And then put the slide back down on the stain tray. Now, your next and final stain for this little experiment is to apply methylene blue to your smear. And you're going to apply this to your smear for about 30 to 45 seconds. Now remember, methylene blue is your counter stain. Now I'm just going to speed this up a little bit, but put that back down, and once you've done that, pick up your slide using your slide clip, tilt it down, and now wash off the excess methylene blue stain with water. Rinse it off gently, and put this back down, the stain tray. And then once you've done that, you can now actually blot dry your slide off. So, I'm going to pick up this, and to make it easier, I'm going to pick this up, my slide clip, and then I'm just going to put it in my book of bibulous paper and just gently rub the slide. And you want to do it until it's completely dry. And once it's dry, you can now view your slide under the microscope. Once you have viewed your slide under the microscope, just put it back in the slide box. Remember, after viewing and analyzing the slide under the microscope, take your slide and put it back in the slide box, which we just did. And then any liquids that were in the stain tray, you're going to pour into the stain collection vessel for disposal. And then you would move all your materials out of the way and disinfect your workspace. As always, you would use germicide to wash down your tabletop, and then you would use paper towels to wipe it up and dry it off. And of course, you would throw any used paper towels away in the biohazardous waste container. And then after you've disinfected your workspace, you would then remove your soiled gloves and put them in the biohazardous waste container as well. Then wash your hands again thoroughly. And then once you've done that, put on a pair of new, clean gloves, because the next experiment that you will be conducting is the endospore stain.